Hi guys, and welcome to our module on hematopoiesis. Now, hematopoiesis or hemopoiesis refers to the formation of blood cells. This would include the red blood cells or erythrocytes, the white blood cells, which includes the monocytes, the lymphocytes, neutrophils, eosinophils, basophils, and macrophages, and then finally platelets, which we typically tend to call thrombocytes. Now, based on the particular type of red cell, hematopoiesis tends to be classified either as erythropoiesis, which involves the formation of red blood cells, leukopoiesis, which involves the formation of white blood cells, and megakaryopoiesis, which involves the formation of platelets. Now, we'll talk about each of these different forms of hematopoiesis in separate modules. We'll run into the details of each of them in terms of their processes, outcomes, and duration. Hematopoiesis as a process tends to occur at varying sites depending on age. In the intrauterine life, there are about three major sites where hematopoiesis tends to occur. In childhood and in adulthood, we have hematopoiesis occurring largely in the bone marrow but at different parts of the bone. So we'll talk about that in the next slide. In the intrauterine life, which is life of the fetus, from around the first to third month of intrauterine life, hematopoiesis tends to occur within the yolk sac of the fetal structure. Then from the fourth week of development all the way down to the fourth month, the liver and the spleen takes control of the process of hematopoiesis. Then finally, from the fourth month till birth, the bone marrow would be the primary site of hematopoiesis. This doesn't mean that other sites don't take a bit of function in the hematopoiesis, but for each of these different phases of intrauterine life, we have each of the different organs taking control, being the predominant site of hematopoiesis. In childhood, whereas hematopoiesis is found largely in the bone marrow, continuing all the way down to adulthood. But this depends on whether we're talking about a child below the ages of 2 years or above the age of 2 years. Between birth and the second year of life, on 2 years of age, the bone marrow of all bones will be involved in the process of hematopoiesis. Whereas in from the second year of life all the way down to the 18th year, which is adulthood, the bone marrow of the long bones, of the sternum, of the ribs, the vertebrae, skull, the sacrum, and the pelvis are the particular bones involved in the process of hematopoiesis. The mechanisms that lead to the transfer or site of hematopoiesis from the yolk sac to the liver to the bone marrow usually are not necessarily known. However, there are bit of an hypothesis that tends to um, states that bone marrow cells tend to secrete some cytokines, including neurokinins and protein P, and these cytokines tend to attract or drive hematopoietic cells towards themselves. So, from going from the fetal stage to childhood, the bone marrows start to develop at that point and secrete those cytokines, and those cytokines tend to draw hematopoietic cells towards them to begin the process of hematopoiesis, making the bone marrow to be the dominant site in childhood all the way up to the adult stage. In adulthood, from 18 years and above, the bone marrow of the vertebrae, of the skull, of the sacrum, and the pelvis are the major sites of hematopoiesis. If you notice that the long bone is no longer involved in the production of cells, blood cells at this stage, that's because the bone marrow within long bones tend to become converted into fatty tissue, what we call yellow marrow, and as a result, that prevents formation of hematopoiesis or hematopoietic cells within the bone marrow of those particular set of bones. Approximately 6 billion hematopoietic cells are found per kg of body weight per day, and usually, in terms of how production of these cells occur, 2.5 billion thrombocytes are produced per kg per body weight in a day. 1 billion leukocytes is produced per kg per body weight in a day, and then about 2.5 billion red cells also produced by per kg by body weight in a day. This is to show how active the bone marrows of these various sites are. Either we're talking about childhood all the way down to adulthood, the bone marrow is extremely active to the point in which millions or billions of hematopoietic cells are being produced every single day. So this is a nice segue into talking about the bone marrow. Bone marrow being the primary site of hematopoiesis from around the fourth month all the way up to adulthood and beyond. it's important that we talk about the bone marrow. So the bone marrow is a spongy, soft tissue that is found within the space between opposing endothelial surfaces of bones. So every single bone has a cavity, what we call a bone cavity. Within that bone cavity lies a marrow, which is what we call the bone marrow, the soft, spongy tissue. And the bone marrow by itself is not the um, large site for production of cells, blood cells. There's a portion of the bone marrow that is involved in the production of blood cells and is involved as the primary site of hematopoiesis. And that particular part of the bone marrow is known as the hematopoietic inductive environment, or the heme. And we'll talk about that very soon in terms of what the heme contains and why it is so unique in the production of blood cells. When talking about heme, it's important to note the connection between heme and the vascular circulation. How exactly do we have cells being formed in newly formed from the heme and eventually being passed down to the circulation? So the connection between that is in the blood supply to the bone marrow. The heme is within the bone marrow, and the bone marrow is supplied by a number of neurovascular structures. The vascular structure of blood supply to the bone marrow goes by, first of all, the nutrient artery. Nutrient artery is gotten from arteries coming from different sites in the limb and the trunk. Nutrient arteries will penetrate into the bone to become what we call medullary artery. But medullary arteries will be found within the medullary cavity, and then they form branches which could either ascend up or descend down the medullary cavity. So we have what we call ascending and descending arteries being the offshoot of the medullary artery. Then ascending and descending arteries eventually become what we call arcuate or transfer arteries. This would eventually divide into smaller set of arteries or capillaries, what we call avician capillaries. I'll show you that in a minute. The innervation capillaries would eventually become even very smaller forms of vascular structures, what we call bone marrow sinusoids. These bone marrow sinusoids is what tend to give rise to the hematopoietic inductive environment. The bone marrow sinusoids are found within the bone marrow and they are in direct contact with the hematopoietic inductive environment or heme. Cells being produced in the hematopoietic inductive environment will be transferred into the bone marrow, and from the bone marrow they can pass through the circulation back. To complete this entire system of blood supply to the to the bones and the bone marrow, the sinusoids eventually become medullary veins, and then the medullary veins eventually become the emissary veins, which leaves the bones to join the larger vascular supply of the particular region where the bone is found. This is a picture showing us the breakdown of some of the branches of the blood supply to the bones. You can see the nutrient artery penetrating through the bone to become the medullary artery in the medullary cavity. Some medullary arteries will be ascending, some will be descending. The ascending and descending ones are called the ascending descending branches of the medullary artery. And when they get to the metaphysis and epiphysis, they become what we call epiphyseal arteries or veins, what we tend to know as arcuate arteries or veins. And then this would penetrate through the bone to become the avician canal. I'll show you pictures of the avician canal and the avician capillaries in the next slide. So this is a picture of the avician capillaries. You can see that the avician capillaries are part of what we call the avician canal. These are smaller microscopic view of the blood supply and the architecture of a bone. And each of the different architectures of the bone have blood supplies to them, which we call the avician capillaries. Avician capillaries would eventually leave the cortical part of the bone to enter into the medullary cavity, then eventually becoming a bone marrow sinusoid, which is what we mentioned is in direct contact with the hematopoietic inductive environment. So let's focus on the hematopoietic inductive environment. What you need to know about this particular part or site or structure in the bone marrow is that it is made up of three major parts. It's made up of an outer surface, also called the luminal surface. It's made up of an inner surface called the abluminal surface, and just medial or within or internal to the inner surface, we have what we call the heme space, the hematopoietic inductive environment space, which is the major site of production of hematopoietic cells. This is a picture of the heme. The outer surface, like I said, is also called the luminal surface. The inner surface, just medial to it, that's the abluminal surface. And then finally, the space, this environment here is called the heme space. The heme space contains a number of things. We'll talk about each of what they are formed by and what they are involved in doing for the entire purpose of hematopoiesis. Let's start with the outer surface. The outer surface is lined by endothelial cells. The inner surface of the hematopoietic inductive environment is lined by a number of cells, and this is helpful for the process of hematopoiesis. The cells that tend to line the inner surface of the hematopoietic inductive environment includes reticulum cells, fibroblasts, adipocytes, and macrophages. Please take note of each of the cells. These cells would all contribute to the formation of structures within the heme space. The outer and inner surfaces are separated by a very thin subendothelial structure, which is made up of a combination of two proteinous material made up of laminin and elastin. The heme space is composed of two major things. The heme space contains extracellular matrix. This extracellular matrix is usually gotten from cells found in the inner surface or the abdominal surface of the hematopoietic inductive environment, whereas the second structure found in the heme space are hematopoietic cells, and these are the cells involved in the process of hematopoiesis. So hematopoietic cells within the heme space are cells that are involved in hematopoiesis, as I mentioned initially, and these cells tend to be organized in a particular pattern that we need to remember. So when we're talking about the heme space, from the heme space going towards the abdominal surface, the cells tend to arrange themselves as cells involved in lymphocytopoiesis, cells involved in granulocytopoiesis, and then cells involved in erythropoiesis and megakaryocytopoiesis. So practically, cells that will become white blood cells are found more towards the center of the heme space. Cells that will become granulocytes will be found more towards the middle between the abdominal space and the heme space. And then cells that become the erythrocytes, red blood cells, and platelets, the megakaryocytes, are found very close to the abdominal surface, and that's what this particular slide is describing. Second component of the heme space is the extracellular matrix. This is derived from cells that are gotten from the inner or abdominal surface of the hematopoietic inductive environment. So remember that this inner surface comprises of reticulin cells, fibroblast, contains also stroma cells and macrophages, and each of these cells give rise to matrix comprising of a different form of structure. So for example, the structures that give rise to the matrix, the extracellular matrix, includes glycosaminoglycan to cement, reticulin, collagen, some growth factors, and then some protein. So the cement or glycosaminoglycans is usually formed by all of the cells in the abdominal surface. So from the reticulum to the fibroblasts, the stroma cells to the macrophages, they all give rise to the glycosaminoglycans. Examples of some of these glycosaminoglycans or cement materials include hyaluronic acid, heparin sulfate, chondroitin sulfate, thematin sulfate, and these are structures that tend to be involved in giving the shape to the heme to the heme space. So they help in forming the shape of the heme space. Then the second is reticulin. Reticulin is formed purely or exclusively from the reticulum cells, and the function of reticulin in the entire extracellular matrix is to give the shape as well, but it also tends to serve as a form of adhesive structure and interaction for immature cells. So immature hematopoietic cells found in the heme space tend to attach directly to reticulin and tend to adhere there to maintain their stay within the heme space. The third important component of the extracellular matrix is collagen. Usually, we tend to see type 1, type 2, and type 3 collagen within the heme space. It serves also as a form of adhesive structure and for the interaction of immature hematopoietic cells. Then we have growth factors like EGF, that's erythrocyte growth factor, thrombo, thrombocyte growth factor, TGF, CG, then GCSF, and GMCSF. So those are granulocyte colony stimulating factors and granulocyte and monocyte colony stimulating factors. All of these factors tend to be found within the extracellular matrix. The function of the growth factors there is to aid the growth and maturation of hematopoietic cells, and that's majorly their function. Growth factors are formed by cells of the abdominal, all of the cells in the abdominal surface. Collagen, I didn't mention that initially. Collagen is formed by cells including fibroblasts and stroma cells. They're not really formed by other cells, but just those two. Then finally, we have proteins found within the extracellular matrix. We have a number of important proteins to note. Some of the proteins found in the extracellular matrix includes the fibronectin, humonectin, fibronectin, vetronectin, laminin, tenacin, endoglin. These are all examples of protein that are found within the extracellular matrix. The function of the proteins in the extracellular matrix is to help to promote hematopoiesis by retaining immature cells by helping to retain. So although these immature cells are already adhering to reticulin and collagen, proteins usually tend to make those immature cells to stay longer within the immune space for maturity to occur, for hematopoiesis to be completed, and then eventually they can be moved into the circulation. The particular cells that tend to give rise to proteins are usually all of the cells of the abdominal surface. These are the cells involved in function of proteins within the heme space. Let's finish this module by talking about the process of hematopoiesis. Just a bit of run through. We'll use this as our template in talking about the different classes of hematopoiesis, from erythropoiesis to megakaryocytopoiesis to leukopoiesis. So starting with all cells or all cellular elements of blood usually tends to be derived from one single set of cells, and these cells are called pluripotent cells. So the form or the starting point of the hematopoietic cells in the heme space within the bone marrow is what we call pluripotent cells. These are cells that can undergo cell division and transformation into any form of cell in the body. So pluripotent cells that we tend to see being involved in hematopoiesis tend to undergo three stages of development during the process of hematopoiesis. These three stages of development includes: number one, the cell stage development; two, progenitor stage development; and the precursor stage development. We'll talk about each of these and what it means for the pluripotent cells to go through these particular stages of development. Let's start with the cell stage development, the stem stage development rather. The stem stage development is the first stage of hematopoiesis for any form of hematopoiesis, whether we're talking about erythrocytopoiesis or megakaryopoiesis or leukopoiesis. Every single one of those forms of hematopoiesis will first start with the stem stage development. Usually, cells within this stage are made up of um, stem cells that have two major properties. They have the function of cell renewal and the function of cell differentiation. So cell renewal is the ability of a cell to undergo mitotic division and its capacity to renew itself. So you have a cell that undergoes mitotic division and does not differentiate, just remains the way it is. So that cell just makes a replica of itself. Whereas cell differentiation is the ability of a cell to undergo division and eventually mature into a newer or um, higher level of cell based on functionality. So stem cells tend to have these two properties: cell renewal and cell differentiation. One important thing to note about all stem cells, is that they are not identifiable morphologically. We can identify them by just looking at them under the microscope, but we can identify them by presence of some markers or antigens on their cells or cell membrane surfaces. So all stem cells in the stem cell stage tend to have a CD34 marker and a stem cell factor marker. They usually CD34 and stem cell factor positive. And this is one way we can identify them, though we can't identify them morphologically. The second stage of hematopoiesis is the progenitor cell stage. This is a stage that involves cells called progenitor cells. Progenitor cells have the ability to undergo cell division and cell differentiation, but cannot undergo cell renewal. So when we have a progenitor cell undergoing cell division, that cell would eventually undergo maturation into a newer form of cell. So they can't renew themselves, they can't replicate themselves and just remain that way. So they must always undergo maturation. And that's one key difference between the progenitor cells and the stem cells. Stem cells can undergo cell division, cell renewal, and cell differentiation. Progenitor cells can only undergo cell division and cell differentiation. They cannot undergo cell renewal. They are also not morphologically identifiable, and unlike the stem cells, progenitor cells tend to be CD34 positive but lack a stem cell factor, and this is very key to note. The reason why we see it's the stem cell factor in the first place is because these are found on stem cells. So if stem cell factors are not found on a particular cell, these cells are likely to be progenitor cells or precursor cells and not stem cells. So the last stage of hematopoiesis involves the precursor cell stage. This contains precursor cells that are committed to a particular cell lineage. So for example, we have cells that would undergo the stem cell stage and then eventually the progenitor cell stage. And when they get to progenitor cell stage, they form precursor cells. Precursor cells are dedicated to forming just one type of cell. So if a precursor cell is for a red cell, that's a red cell precursor. It will always give rise to a red cell. If a precursor cell is a platelet, it will always give rise to platelets. If a precursor cell is is a granulocyte to always give rise to a granulocyte. So these are cells that are committed to a particular cell lineage. There are cells that cannot undergo cell renewal, similar to progenitor cells. But unlike progenitor cells, they are morphologically identifiable. So when we view them under the microscope, we can identify precursor cells and know what particular stage those precursor cells are in, and depending on what particular type or classification of hematopoiesis we're talking about. So this is just a run through of the process of hematopoiesis. You could see the stem cells becoming what we call progenitor cells. We have the myeloid progenitor cells. These are cells that will give rise to red blood cells, platelets, and granulocytes and monocytes. Then we have the lymphoid progenitor cells, cells that are committed to the formation of lymphocytes. Then the progenitor cells will eventually give rise to precursor cells. You can see each of the precursor cells there and the specific cells they give rise to. So myeloid progenitor cells would either give rise to megakaryocytes, eosinophils, basophils, erythrocytes, monocytes, and neutrophils. These are all precursor cells, and this is their adult form at the end of the day. And finally, for lymphoid progenitor cells, they give rise to precursor cells that are either T cells, B cells, or NK cells. And this is practically the overview of hematopoiesis. We'll talk about hematopoiesis in specifics in a different set of videos. So we'll talk about the erythropoiesis as a separate video. Same thing with the megakaryopoiesis, lymphopoiesis, and granulocytopoiesis in a separate video, in a separate model. And that concludes our module on hematopoiesis.